From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 5, recorded on April 15th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from New York City, Lieberman.
Hi, Vincent.
Also joining me from Salt Lake City, Jason Shepard.
Hi, Vincent.
How are you doing?
Good. Yeah, it's spring here, so it's I'm itching to get outside, but it's nice. We can still climb the hills and mountains by Salt Lake City, which is a good thing.
Good. Also joining us from New York City, Bendesky.
Hi, Vincent. Hi, everyone.
From Nashville, Tennessee, Erin Calipari.
Hi.
Welcome. And our guest today from Boston, Massachusetts, Bob Datta. Welcome.
Hi, Vincent.
Well, amazing. We have a full house here, plus one. Bob, tell us a little bit about yourself.
What do you want to know?
Where are you from?
I'm originally from Long Beach, California, but I spent most of my life on the East Coast ever since I went to college.
Where'd you go to college?
I was an undergraduate at Yale.
Uh-huh. And were you an MD or PhD or both?
I'm both, although I never trained as a doctor, so I'm an honorary MD.
Uh-huh. Where'd you get your PhD from?
I went to Harvard Medical School, and I was a graduate of the Department of Neurobiology, in which I'm now a faculty member.
I see. Very good. Is everyone doing well? Let's hear from each of you. Ori, how are you doing?
I'm good. I'm quarantined, I guess, as is everybody. So we're out of clinic and had my exam today, and kind of just planning to do some board studying and cooking a lot, occupying my.
Yeah, you've been on TWiV a couple of times and told us about no class.
Yeah, it's very odd, but can go to that TWiN crossover and take a listen, I guess. You're not
a senior, right? You're not graduating?
No, so all of my MD PhD friends who graduated essentially a year faster than me are graduating now today. Yeah, and are starting in. Sorry, my cat just popped in.
It's okay.
And they're graduating today, and some of them are starting in the hospital within a week, I think.
Yeah, they accelerated, right?
Yeah, so they'll mostly be doing kind of more administrative tasks, and then we'll all start in July for their intern year.
You know, cats can be infected with SARS-CoV-2.
I know. I'm worried about them, but they're, as always, in quarantine and self-isolated. So.
Yeah, Ori and I are in the same institution. Well, Andres as well, but we shut down a while ago. But if you're doing SARS-CoV-2 research, or if you're essential, you can go in. I've been going about once a week to change the CO2 and nitrogen because no one else can do it in my lab. So, but otherwise, I'm here doing all sorts of things. Andres, you said you're going in a couple of days a week?
Yeah, same as you. I guess I go take care of our animals, make sure they're okay. They have food, they have water, they're healthy. Yeah. So essential work, yeah.
Yeah.
How has it been in New York? Is it bad? Is it worse than other places? I guess like down here, it's kind of, not great, but we're spread out. In Nashville, people aren't on top of each other. I did my postdoc in New York, so it's it's easier to social distance down here, and the houses are a little bigger. So when you get stuck, there's not like just one spot. So how are you guys holding up?
So Ori, what's the medical situation there in the city?
So the city is quite bad. There was it peaked about 3 or 4 days ago, and the hospitalizations were going down, and then went back up today and yesterday. And there are something like 800 people on ventilators in Columbia and New York Presbyterian. So it's a tremendous amount. I was talking to a friend yesterday, but it's interesting. I feel like things are a little bit more normal, but I think it's because we're getting used to this very strange reality that we're in, not because anything has actually improved. But if we record long enough, we'll hear people cheering at 7. So,
What happens at 7?
everyone kind of sticks their heads out the window and cheers for the medical workers.
Oh, I didn't know that. Oh, interesting. That's cool.
Yeah, I live right next to Mount Sinai in Morningside, and the fire department, the fire truck comes, and they turn on their sirens, and all the neighbors come and bang on pots and pans and for 5 minutes.
That's cool.
It's pretty encouraging. And the doctors and nurses come out and to be clapped.
Jason, what's it like in Salt Lake? You have a lot of cases there?
No, it's actually been not too bad. The surge that they predicted never happened. In fact, I have an ER doc friend who got sent to California because he wasn't needed here. So that's promising. And we'll see if it stays that way. I think same as Nashville. Salt Lake's a little more spread out, and everyone in Utah pretty much lives in this valley. So it's a little easier to social distance, but there's been some rumblings. I mean, we still have rumblings of people trying to, go to church and that sort of thing. So we'll see if this, the curve is flattened as much as we hope. But otherwise it's not too bad.
How about you, Bob? And what's Boston like?
Yeah, we're in the middle of our surge right now.
Yeah.
We were a little bit behind you guys in New York. And so I think we're right in the thick of it currently. The big news, I think, in the last day or so was that they screened everyone at one of the biggest homeless shelters in Boston, and a third of the people who were living there were positive.
Wow.
For COVID. Almost entirely asymptomatic, but a huge number of potential carriers. And so overnight, they got those guys isolated and in safer housing. But I think it's emblematic of the problem that faces us here and probably in New York and lots of other places.
It would be great if 30% of the U.S. were infected, but I think that's a special population because they're all in the same shelter together, right?
Exactly.
Yeah.
It seems like there's really a lot of asymptomatic folks, right? I mean, there was this New York study from pregnant women coming in where 80% of them were asymptomatic. So if that's true, we're certainly probably underreporting infectious cases.
I hope so.
In that study, though, it was a little bit. So they were 80% that were asymptomatic on admission,
right?
But then only 30% remained asymptomatic through the labor and delivery.
30% remained. Okay.
Yes, many of them developed coughs and fevers after birth.
But even 30%, that's still pretty high, right?
Yeah, and many of them actually decompensated quite rapidly in labor. And so there was kind of this push to say, even though pregnant women are scared to come to the hospital because of COVID, they should come because you don't know whether you're infected, and delivering at home can have very bad outcomes. So it's kind of a public health thing. Yeah, it's very interesting.
And Erin, you said it's pretty quiet down there in Nashville?
Yeah, but that could be because we shut down pretty early. Davidson County and Nashville shut down kind of like when the coast did, but nothing around us did. So I don't know if it's actually quiet or we're living in this little bubble of like none of our neighbors go out. Vanderbilt's been shut down for a long time. So we're just going in and keeping mouse lines alive and cell lines alive. But we've been shut down for a while. But we haven't really seen like a massive surge here either. And for us, Vanderbilt's kind of the major medical center for a very large area of this country. Like, in New York, it's so different. You have all these hospitals in such a small area. Down here, you have like Vanderbilt. Yeah. And that's it. And everybody from a very large radius comes to Vanderbilt. So, I think Davidson County that we're in is going to see the worst. Like my zip code is the worst for cases, which is actually kind of weird because we have interacted with nobody. So it's kind of interesting to see how it's playing out because, when you don't leave your house, it's a little hard to get a good kind of vibe of the area.
I did you have a terrific coronavirus guy there, Mark Dennison. I don't know if you've heard, I did a TWiV with him a couple of weeks ago. He's into antivirals and so forth. And that was really good.
Yeah, it's been kind of cool to see, the kind of different facets. I'm in such the neuroscience side. And so I've been here a little while, but I'm still kind of learning. And so when these kind of different things pop up, you get to learn about your institution and all these people doing really great work around. Cool. And I've seen them, they're all over the place, like on CNN. I turn on the TV, there's always like, all the people doing the COVID work there. So it's kind of neat to see these other people at your institution and like what they're working on. So that's been silver lining.
We have had dozens of labs all of a sudden decide they want to study this virus. I feel I've been ignored for years, and now finally virology is popular again. So, but not for a good reason.
To see people do that, right? It's like, for me, I'm like horrified to do something in my area that's remotely related because I feel like I'm not going to do it well enough. And then some people are like, I've been studying systems neuroscience, but now I'm going to do virology. And I'm like, go for it. Like, I guess.
Well, there's been an interesting debate about this on Twitter. And some people are saying, no, just let the experts do the thing. And then others are saying, well, we should all be contributing. But I think it comes down to like, can you guarantee that your trainees are going to be safe when they go to the lab and that sort of thing, and what's the cost benefit there? And I'm a pseudo virologist right now, and so even my labs, though we're shut down, trying to figure out, is there anything that we can do right now that can make an impact? And I'm not sure.
Yeah.
Well, I think also too, if you're at a place where there's infrastructure, where there are experts in certain areas around that you can kind of synergize with, I think that's where there's a lot of potential. I think there's, it's kind of hard, right? If you don't have anyone around you that has any experience in any of this, and you say, I can figure it out, that's a totally different ballgame than a kind of similar research area or a tool that clearly is applicable, and then getting somebody at your institution or close that has that experience and synergizing where there's a problem is, I think, a really great way to do it. But I feel like that's like thinking about how it's applied is really the important thing when people start switching areas, rather than just going in and saying, we'll get data and figure it out. There's all those nuances and fields that are kind of different. And so having people in there is, I think, an important
So,
thing.
Ori, you want to start out with a little overview of SARS-CoV-2, and then you can talk about neurological manifestations?
Yeah. So, do you want to walk through a little bit of the virus life cycle together? And then, or maybe we should just say that for this episode, we'll talk about kind of neurological manifestations of COVID, including changes in taste and smell and kind of other CNS manifestations. So maybe, Vincent, do you want to tell us just a little bit about what this virus is?
Yeah, sure. And in fact, this is a good group to talk about the neurological manifestations. It's pretty interesting. I did a TWiV yesterday. Someone said SARS-1, there were some neurological manifestations, but quite rare. So I don't know if we've seen it yet here, but you can tell us later. So this is a coronavirus where we know a whole bunch of coronaviruses that infect humans and cause a spectrum of respiratory disease from common cold. There are these seasonal endemic coronaviruses. There are 4 of them that cause mild disease. Then we have these epidemic coronaviruses, SARS-1, MERS coronavirus, and now SARS coronavirus 2. They can cause a wide spectrum from pretty mild upper tract disease to acute respiratory distress, requiring oxygen and intubation. Of course, that's the problem. We're putting a stress on the hospital system. And I don't know if you've noticed, Ori, now the picture has turned slightly. There seems to be a coagulopathy in a lot of patients that's leading to oxygen deprivation. Is that right?
Yeah, that's what kind of the word on the street is. And there have been some perspectives in, I think, JAMA and other kind of internal medicine journals suggesting that early anticoagulation is helpful to prevent kind of the later sequelae of the disease. Mm-hmm. And some of the neurological manifestations seem to be secondary to this coagulopathy as well. So I guess we can transition into this paper, the first of kind of 2.5 papers we'll talk about today. So this is a paper that Jason sent out about in JAMA Neurology about the neurological manifestations of hospitalized patients with coronavirus disease in Wuhan, China. The first author is Ling Mao and the last author is Bo Hu. So this is a retrospective study. So they went through all of the charts of COVID patients. They analyzed, I believe, 214 patients that are in order. So these were 214 coronavirus positive patients who were admitted sequentially to these hospitals. And then they chart reviewed and looked for neurological manifestations, and they found a third of patients had neurological manifestations. These were split into 3 categories. So CNS manifestations, so change in consciousness, late state of consciousness, headache, dizziness. cerebrovascular disease, so kind of getting at what Vincent said about the coagulopathy, or seizure, peripheral nervous system manifestation, so taste and smell impairment, which we'll talk about a little bit later, and then skeletal muscle injuries, which are kind of in the boat or the wheelhouse of neurology as a kind of a medical specialty, but in my mind is really a separate kind of idea from nervous system involvement. So one question that they talk about in this data set is, at what point in the hospital course were these neurological symptoms present? And these were mostly actually present early in the hospital course. So you could imagine that this could suggest this might have more to do with a part of the disease where the virus is actively replicating, although we don't know how long these patients were sick at home before admission. The second thing they looked at was severe versus non-severe disease. They separated patients based on standard methods to rate severity, and they saw that neurological manifestations were present more commonly in severely infected or affected individuals. And finally, they talk a little bit in the discussion about how this fits into our understanding of coronavirus biology, and compare a little bit with SARS and MERS, SARS-1 and MERS. And there's some interesting data that I hadn't seen, and Vincent, you should tell me what you think about this, that, for example, there has been isolation of SARS-CoV-1 from the CSF of a patient, and that there are some cases where in autopsies or postmortem studies that there are changes in the brain, so cerebral edema, neurodegeneration, axonal degeneration that could be associated with viral infection or just kind of the immune response afterwards as well, or just being very sick. So this is just to me kind of raises awareness, because I don't think I had this in my mind as part of the COVID kind of disease spectrum. I reached out to Daniel Griffin, who's a frequent TWiV guest, who is an infectious disease doctor on Long Island. And he said that he's actually seeing this in many patients as well. They started to call this a COVID-19 encephalopathy. Mm-hmm. They're seeing it in later parts of the disease, though. So you could imagine that someone's in the ICU, they're lying down, they're infected. This is a perfect time for there to be coagulopathy and kind of general problems of severe illness, and then they're having neurological manifestations. And I think that just more data will help us understand what is going on here.
Yeah, these things seem like all secondary effects to infection, right? Rather than virus in the CNS.
But the encephalitis, I mean, could you get encephalitis from brain, cells sort of getting infected in the brain itself?
So I don't know if there's I don't know if there's data of CSF inflammatory cells in the CSF in these papers. This was mostly just describing symptoms, in which case you could imagine just like systemic inflammatory responses causing neurological manifestations. It would be interesting to look at CSF cell counts and see whether, or for that matter, viral PCRs, and see whether there is viral infection or viral RNA within the CSF.
Yeah.
And we'll get to this with Bob's data on how it's not particularly neurotrophic. The receptor is not expressing most neurons, right? So if there was some encephalitis, it would be indirect through the vasculature or secondary process to coagulopathy or something else. It seems unlikely that it's infecting neurons directly like herpes or other viruses.
But this, Ori, you're not likely to do a spinal tap on these patients, right?
Exactly. Especially, this was obviously a retrospective study, so there most likely were not spinal taps.
Yeah.
The question is, if this is really going to become a thing, there can be prospective studies, and people can now start to think about that.
Well, I think one of the worries here is that the long-term consequences of infections and whether there's any sort of neurodegenerative component to it, something similar to HIV.
So, Ori, do they have any sense of how this contributed to demise of these patients? I assume a certain fraction of them died, right?
Yeah, I'm trying to look at what the number who died were. They didn't comment. They didn't comment on that. So Vincent, you're an expert on kind of a virus entering the nervous system. So does this make sense to you in terms of kind of moving from the gut to neurons, or kind of an analogy to polio?
So you could get from the gut or the respiratory tract to the CNS. I mean, polio goes from the gut, of course. Measles goes from the lung to the central nervous system. It causes encephalitis in about 1 in 1,000 kids with measles. So there is a route. This virus Entero68 that we work on now goes apparently from the lung to the CNS, causes acute flaccid myelitis. So yes, there are pathways. But as Andres said, if there's no receptors on neurons, well, I guess it could infect. How about astrocytes, Andres? Do you know?
Maybe I think Bob has data on all of the, so maybe once we get to his paper,
we'll wait for that.
More about it.
Yeah.
But also, I mean, I'm curious to know if about co-receptors as well. It's not just ACE2, right?
Seems to be ACE2 at the moment. Yeah, that's all we know about. If you put human ACE2 into mouse or other cell types, it will allow replication. So that's all we know right now. Interesting. I'm curious, Erin. I mean, you're the person to sort of address this. I mean, what's the psychological implication of a pandemic like this and making people stay at home? Does that have implication?
Well, so two things. That's actually a huge deal, right? Because you talk about what the virus does to people. So that's the first angle, but then also what social isolation does to people. Yeah. So you have people who have pre-existing conditions like anxiety, depression, these kinds of things make those worse. Yeah. And then also, I mean, the other thing that I study is drug addiction. And so when you have people who are isolated in their homes, you have all kinds of increased incidences in things like overdose deaths. You have increases in stress-induced relapse and things like this. And so this kind of expands beyond just what does the virus do and goes kind of into like the societal implications of this added stress, along with social isolation and lack of kind of human interaction and support systems. And a lot of these people with these problems like addiction or depression or anxiety, a lot of them are already socially isolated. So you further isolate them from these kind of networks where they're interacting with people, and now they live alone and they're literally home all day by themselves. And so I think this has implications too for what we're going to see when we come back out of this, where we're going to probably see a spike in problems with psychiatric disease as well. Yeah. So, it's kind of, you have the first pass, which is what's the virus do, but the next pass is like, how does this societal change affect people and how's that going to affect things in the future? I think the big thing is this has implications like far down the road as well, which is, kind of scary. And if we have the infrastructure to kind of deal with these issues, I don't know, because I think we haven't really gotten there. We're still at stage one, which is how do we stop this problem, and not kind of how do we clean up all of the things that this caused.
Good point. There's also, there are many impacts not directly related to virus replication, like the economic impacts are huge, right? On top of that, it's all kinds of wrecking of industries and so forth. And I'm sure there are many others that we haven't touched on, but and I suppose individuals who normally see someone regularly for their problems, their addiction or psychiatric problems, they can't see them anymore, right?
Well, especially a lot of these things that what we deemed essential and what we don't. It's kind of an interesting thing, right? Because we've gone to telemedicine for a lot of stuff, which I think is actually really great because it's showing us how we can efficiently kind of mobilize a system that kind of reaches far into kind of maybe remote areas. But also you lose that. I mean, we see Zooming is not the same thing as human day-to-day interaction.
No, it's not.
So I think that when we've kind of picked up in maybe efficiency, we lost that kind of human component. I mean, it's still, we're doing our best here, but I think that a lot of those things too, compound and they're limited, right? You have, let's say we Zoom for this like hour, but then it's over, right? Like day to day at work, even if you kind of work a little bit alone in a science lab, you still have like those day-to-day interactions and people say, hey, how's it going? There's a change of scenery. And I think a lot of your scenery at home is going to depend on your socioeconomic status as well. So when people lose their jobs and you're talking about an added stress there, or even thinking about essential workers who have to go to work, that is stressful for them as well. So each person now is experiencing the stress they didn't. There's probably going to be manifestations of that for new diseases or new problems that people haven't had before. and exacerbation of problems people did have before and limited treatment and access. And it's a big problem that I think we're just starting to think about how we would address that. I don't know if we have the infrastructure right now to do it in the time frame we're going to need to do it in.
And we have a new faculty member, Moriel Zilikovsky, who's been doing studies on self-isolation or isolation, the effects of isolation. It's actually astounding to see what effects, long-term effects there are. She's done all sorts of different behaviors from aggression to, sexual behaviors, and they're all affected by social isolation.
Oh yeah. Especially in animals that are not normally isolated. So I did a lot of social isolation work in grad school, and there's, the transient effects of isolation that happen during, there's the long-term effects. And then also there's a developmental angle about, effects on adolescents are way more robust and way more long-lasting than isolation effects on adults. So you also have that added component of how taking maybe children out of their kind of network, how that disproportionately affects them relative to adults, and that other component, which is even more complex. And so,
all the schools are closed, and even Utah closed their schools for the rest of the year. So crazy.
It's remarkable. Yeah. Well, maybe we can move on to Bob's paper. What do you think, Ori?
Yeah, that sounds great.
So, Bob, you have a bioRxiv preprint. Yeah. Non-neuronal expression of SARS-CoV-2 entry genes in the olfactory system suggests mechanisms underlying COVID-19 associated anosmia. Is that how you say it? Anosmia?
Anosmia.
Oh, tell us all about that, Bob.
Sure. So, as I think was mentioned a little earlier, it seems anecdotally and increasingly now in formal reports that COVID-19 is associated with changes in smell and taste. There have been just kind of surveys that have gone out of doctors kind of collecting clinical data that suggest an unusually, at least what it seems like now, an unusually high rate of people complaining about changes in smell perception or the absence of smell. And hard data is beginning to emerge about the rates at which that's occurring. And it does indeed seem currently like some substantial fraction of COVID-19 patients end up with a change at some point in their sense of smell, often early in the disease, actually. In the tens of percents, at least in the current reports. And who knows whether those numbers will stand as we collect more data, maybe think about things more prospectively and in a little bit of a less self-selected sort of manner. But I think it's clear at this point that alterations in smell and taste are going to be part of this disease spectrum and one of the sequelae of infection.
Do you know how much in people? I know I've heard many anecdotal cases of people, their only symptom is anosmia, and that maybe resolves. But do you know of people who've had more severe disease, how long it persists? Is there more data? Is it chronic? Does it resolve and then it's followed by other symptoms? Or?
So I think we're just beginning to understand this now. I think there are many cases where people's only symptom, at least by self-report, is anosmia. The first numbers about rates of recovery are beginning to emerge too. I think that's just a very important point. It seems like most people who complain about a change in smell or taste end up recovering after they recover from the acute kind of sequelae of the disease. But we don't really, I think, any of us understand fully the natural history of this disease. And so I think, it'll take some time for us to really understand how people are being affected long term. And I think one thing that's probably worth talking about now, because it's not something that I think is fully appreciated, is how profound a chronic or a permanent change in your sense of smell can be for you psychologically. So there's a ton of data actually suggesting that if you chronically lose your sense of smell, it dramatically affects your sense of well-being. We were talking a little bit earlier with Erin about how you require these kinds of day-to-day social interactions to keep yourself psychologically healthy. And it turns out that there's these very intimate connections between the olfactory system and kind of memory and emotional centers in the brain, right? There are two synapses between your nose and your hippocampus or your amygdala. And so it's thought that your sense of smell provides you with a kind of ongoing hedonic tone. And when you lose that hedonic tone, it can really be devastating. And so although we think of smell as kind of the bonus sense, there's a long list of philosophers who just thought of it as dispensable. Actually, there could be some, if this ends up being a permanent thing where some substantial fraction of patients end up permanently losing their sense of smell, this could actually be an important clinical problem going forward. But at least currently, it seems like most people are getting it back, which I think is a good thing.
And I mean, correct me if I'm wrong, Vincent, but this loss of smell is actually not uncommon with viral infections, whether it's long-term or not. But because I actually have a friend that lost their smell for 2 years. And they think it was a viral infection, but sometimes hard to know.
Yeah, it is common with other respiratory tract infections. Years ago, I had a cold and I totally lost it as well, but it came back. So it is not unique to SARS-CoV-2.
One of the challenges here is that we know that if you query people who show up to like an anosmia clinic and you ask them, have you been sick recently? A significant fraction of them are like, yeah, I had the flu, I had a cold, I had some viral infection. We don't actually know the flip side of those data. So we actually, believe it or not, have no idea what fraction of people who, for example, get influenza go on to develop a long-term change in their sense of smell. And so we're seeing these numbers that are 10, 20, 30, 50, 60% for COVID. We actually don't know how typical that is. But it certainly seems like a hallmark symptom of the disease and worth paying attention to.
And in many upper respiratory tract infections, the anosmia is related to edema, inflammation, and fluid that prevents odorants to get to the olfactory epithelium. But in this case, as Bob's paper shows, it's most likely a direct effect on the epithelium rather than fluid or blockage or inflammation.
That was surprising that most of the symptoms are a dry cough and no runny noses, as opposed to the common cold, which is mostly runny noses. And I'm wondering, why is that the case? Why don't you have that kind of response?
No one knows the answer.
Bob, you got interested in this with SARS-CoV-2 because you're interested in olfaction in your lab, right?
Yeah. So actually, Erin was joking around a little earlier about how systems neurobiologists maybe shouldn't become instant virologists, and I'm certainly guilty of that. So actually, in this particular case, I think we were pretty well set up to think about this as a problem. One of the problems that some really great folks in my lab work on, David Brand, Tetsuya Tsukuhara, and Caleb Weinreb. These guys have, for a couple of years now, been really diving deep into the molecular biology of the peripheral olfactory system. And so, when COVID hit, we had a ton of sequencing data in the lab looking at gene expression in the nose, in the olfactory bulb. And these guys had already basically collated these very special data sets that had been previously published from across the world, looking at various kinds of sequencing data in both mice, non-human primates, and in humans. And so we had this kind of like rich data set at both the bulk and the single cell sequencing level that would allow us to kind of look at whether any of these COVID-related genes were expressed anywhere in the olfactory system. And so, when it hit, the lab got shut down. All your labs got shut down too. And we realized that we could answer at least one small question without having to go into the lab physically. And so we decided to jump in and try to answer that one question.
So can you walk us through briefly the anatomy and kind of basic physiology of the olfactory system?
Sure. So I think the one thing that's worth knowing, right, is that your olfactory system kind of has two epithelial components to it in general. There's inside your nose, there's the respiratory epithelium, which is actually continuous with the respiratory epithelium that's in the rest of your pulmonary system. And it has a bunch of cell types that have cilia and secrete mucus. And this is kind of a continuous epithelium with what's in your nasopharynx and what's a little bit lower down in your lung. And then you have a specialized subportion of your olfactory of the epithelia that lives in your nose that's dedicated to olfactory sensation itself. So that's called the olfactory epithelium. And that's the part of the nasal epithelium that houses the olfactory sensory neurons that are responsible for smell. And so the cells that live kind of up in there are related to, but quite distinct from the cells that live in the respiratory epithelium, for example. And so when people started obviously noticing that COVID-19 was associated with pulmonary symptoms, one of the first things that they did was to go look for this SARS-CoV-2 receptor, ACE2, right, in various regions of the lung. And it's clear that certain cell types lower down in the lung and the respiratory system express reasonable levels of ACE2. And interestingly, kind of more upper airway and nasopharyngeal respiratory epithelium also expresses, they has cells that express ACE2. But no one had actually looked specifically at the olfactory epithelia. And so we began to kind of think about what cells might live in the olfactory epithelia that could in principle get infected. And of course, the olfactory epithelia has these sensory neurons, which are responsible for expressing odorant receptors and for detecting smells. These are the cells that have axons that puncture the cribriform plate that sits at the base of your skull. And so their axons terminate in a structure called the olfactory bulb, which is like the first way station. It's kind of the gateway for olfactory information in your brain itself. And so there's almost a kind of continuity between the cells in your nose and their axons and the olfactory bulb at the base of your brain. And those neurons are supported by a wide variety of cell types, including support cells. Some of the most important support cells are sustentacular cells or microvillar cells. These are cells that are responsible for maintaining ion balance, which allows the sensory neurons in your nose to fire. It turns out your olfactory epithelium is constantly bombarded by toxins and infectious agents and debris. And so those neurons get killed just because they're basically facing the airspace and getting kind of pummeled all the time by stuff coming in through your nose. And so there's stem cells that actually live in the epithelium that renew these olfactory sensory neurons on an ongoing basis. And so this olfactory epithelium contains, a bunch of cell types, including the neurons responsible for smell, these support cells, and a bunch of stem cells to continue to regenerate that epithelium over time.
And so what did you find?
And so, I think our naive expectation was that we would find that ACE2 and some of the proteases that are involved in facilitating SARS-CoV-2 entry would be present in neurons, because this kind of acute loss of smell is this hallmark feature of the disease, it seems. But instead, in all of the data that we saw, from the mouse, from non-human primates, from some very special human samples that Brad Goldstein and Hiro Matsunami at Duke had obtained, in all of the data, we failed to identify essentially any ACE2 transcripts whatsoever in the neurons that live in your nose. But instead found that ACE2 seems to be expressed in these stem cells, and especially in these support cells that seem to be involved in setting ion balance, for example, in the olfactory epithelium itself. And so there are definitely cells that live in that epithelium that are capable of being infected. And one of the neat things that Caleb was able to do, because we had access to all this data, was to essentially ask, is the level of ACE2 expression in your olfactory epithelium, is that comparable to the level of ACE2 expression in the rest of the respiratory tract? Because we know the rest of that respiratory tract gets infected. And so if you want to ask questions about plausibility, we have this kind of opportunity to use gene expression to answer that question. And so it turns out that through some kind of clever normalization, that the levels of ACE2 in, for example, the sustentacular cells, which are these support cells in your olfactory epithelium, are pretty similar to the levels of expression of ACE2 in other cells in the nasopharynx. And so we think it's plausible that those cells are getting infected.
So you find that the levels are high in a very small subset of cells, right? Like less than 2%. What do you think? Do you think this is a particular subset of the sustentacular cells or the other epithelium, or what do you think about this low percentage of?
Right. So as you know, Andreas, interpreting single cell data can be challenging because you don't know whether you're undersampling. So one could easily imagine a model in which there are a limiting number of transcripts that are ubiquitous across all the sustentacular cells. We're only able to detect them in a small subset, and that's why it looks like only a handful of cells actually express ACE2. So that's kind of one possible model. There's just not a lot of message, but it's ubiquitous. And the other model is, of course, there's some special subset of cells that seems infectable. And right now, without additional experiments, I think it's hard for us to know. We have been in the lab, and this hasn't made the the paper yet. And there is some data in the paper. We've done a little bit of staining. So Brad Goldstein has some incredibly precious samples of normal human epithelia that he got. He's an MD PhD. He was able, as part of ENT procedure, to go ahead and get biopsies of actual human epithelium. And he had them in the freezer and was able, before his lab was shut down, to stain it with ACE2 antibodies. And in those samples, you can clearly see staining in substatacular cells, and it's maybe a little bit less sparse than we would have imagined given the sequencing data. And so it's possible that some significant fraction of these cells actually, in the end, will be in principle infectable. And we're kind of doing backup experiments now in my own lab. We were given permission to have one person come in a couple days a week just to do some quick ACE2 staining in samples in mouse olfactory epithelium samples that we had in the freezer. And so we're looking at those right now. And our preliminary data, we're seeing very similar things to what Brad saw in his human samples. And so we don't really know the answer, but we think it's not 2% of the cells. It's probably actually more than that.
So would infection of these cells and either destruction of them or perturbation, would that lead to the loss of smell? Does that make sense?
Yeah, I think there's kind of two possible models, both of which are reasonable. The one model, which is obviously kind of intuitive, is that it could just cause a ton of local inflammation right in the olfactory epithelium. So, your nose is 97% respiratory epithelium and 3% olfactory epithelium. We as humans aren't really specialized to smell. It's a sense that over evolutionary time has been progressively waning in our branch of the primate lineage. Nevertheless, it's possible that just local inflammation up at the top of your nose, where the olfactory epithelium lives, could be sufficient to block smells from coming in and contacting the sensory neurons, or the inflammation itself might prevent the function of those neurons. But the other kind of
Go ahead.
Yeah, I mean, the other mechanism, which is much more speculative and kind of interesting, is that actually by disrupting the microvillar cells or the substatacular cells, you might actually change the ion gradients in the epithelium. That might act like a kind of light switch to turn off your olfactory sensory neurons' ability to smell. One of the side effects of the kind of analysis we did was that we discovered that.
Are there known, so Vincent, you can tell me if I'm wrong about the nomenclature here. So this would be viral entry, but then there would be other genes that are required for permissivity of replication, right?
That's right.
So are there is that known for SARS-CoV-2 or other coronaviruses? And can you kind of collate that data with the viral entry data?
Yeah, I think it's not known yet because it's so new, but there were certainly there are certainly internal factors that would control it as well.
So in his paper, Bob does look for this other TMPRSS2, right? This protease that allows for the viral fusion. But Vincent and Ori were talking about more downstream genes that are important for replication or other aspects of the biology.
The virus might not even replicate in these cells that you find positive. It may be in surrounding cells, and then the inflammation could cause the damage, as you say, right?
Exactly. The other thing that's in the paper is that we went ahead and looked at some single-cell sequencing data of the olfactory bulb, and we did some meta-analysis of other people's sequencing in the brain to kind of directly address this question of whether or not neurons themselves can be infectable. And we could not find ACE2 message in any neurons. Interpreting a negative result like that is a challenge, right? But at least it seems like it's expressed at very low levels compared to where we did find it, which is in these parasites, these vascular parasites that seem to be involved in maintaining the blood-brain barrier and clearly are involved in inflammatory responses in the brain. And so the idea that there's something neurovascular fundamentally going on that underlies many aspects of the CNS disorders associated with COVID seems reasonable given our paper. You might imagine that if the olfactory bulb gets inflamed, that you could lose your ability to smell through that kind of mechanism. And of course, there are these holes right at the base of your skull that allow the first cranial nerve, the axons of the sensory neurons, to kind of course up into the brain. And so there's a kind of route of entry for viruses that naturally exists between your nose and the olfactory bulb itself. And so it's possible.
So one of the ways of knowing or figuring more of this, what are the cells that get infected and are leading to the loss of smell, is it possible for you to get biopsies or autopsies from patients with the virus, so you can detect through single cell sequencing what cells have the viral RNA and what type of cell they are? Or
yeah, that's a great question. But from a public health perspective and a practical perspective, that has been a challenge. Getting autopsy tissue on patients like this, given how dangerous the virus itself is, has been difficult. So the very first autopsies are being reported out now. They don't include, as far as I know, any analysis of the olfactory epithelium. Mostly are reporting out that the cell types that you would expect to be kind of altered are in the lung, which is useful information to know from a basic science perspective. But hopefully we'll be able to gain access to actual human autopsy tissue soon.
It's actually been very difficult to get autopsies, even though sadly with so many deaths, because the family are not allowed in the hospital, so you cannot consent them by phone to ask them if you can perform an autopsy. So it's been actually challenging to figure out more of what the disease is. It's a sad consequence of people dying alone.
As far as I've seen, there's like less than 10 autopsy reports that are out there. Yeah.
There's only one for MERS coronavirus.
Only one?
One autopsy. Only one, because the Saudis don't want to do them in general. And so we have a minimal understanding of that disease, even though we get 2 cases a month, something like that, continuously. It's worth pointing out that we used to think polio actually went through the olfactory bulb, and some of the early therapies were to spray things into people's noses to prevent infection. But of course, it didn't work because it gets into the gut. But you don't find the receptor on neurons, so that's an interesting observation. It's not going to go in through those.
And a reason for optimism, I guess, in a certain kind of sense, right?
Yeah,
for sure.
Good news.
So what's the evidence from? So Vincent mentioned if you put the receptor ACE2 in a cell that doesn't have the receptor, and the virus will get in. But if you knock it out from cells that normally had it, will the virus not get in at all? You mentioned this other potential receptor, less efficiency one. But what's the evidence of secondary receptors?
I don't think there is any good. I think ACE2 is, if you put ACE2 in many cell types, you can.
But if you remove it from cells that already have it, does it not get in at all? Or
that's not been done yet. I'm sure it's ongoing. But at least for SARS-1, there was no evidence for a second receptor. You do need this protease, of course, and there's a second protease cleavage site in this spike for furins. And that's interesting because the closest virus, the bat virus isolate, doesn't have a furin cleavage site, and that may have enabled human transmission. But I don't know. Did you look at furin's expression in these cells, Bob?
We did, in fact. And furins are present in olfactory sensory neurons, it turns out. So if you imagine, the premise of our study is that ACE2 is obligate for SARS-CoV-2 entry.
Yeah.
As the science emerges, that if that turns out not to be the case, we kind of queried all the genes that have been associated with SARS entry, including all of the proteases, the cathepsins, and some of them are in fact expressed on neurons in the brain and in the nose. And so this still is to some extent an open scientific question.
So I have a question. So okay. This is like a clear, potential for how it's affecting certain neurons, but not others. And you kind of touched on the inflammation hypothesis and how that could maybe affect the neuronal function of these kind of sensory neurons. But then I guess my question is, why would it be so long lasting, right? But it's not permanent, or maybe it is permanent, right? So I could see a kind of way where you could get inflammation and the cells maybe die or are no longer functional. But these like long-term but still transient changes in kind of olfaction, that sounds, how does that happen? Do you get some sort of like epigenetic plasticity in these cells that lasts for some period of time? Because that would mean that you don't, or do you get cell death and maybe eventually they're replaced? Like, kind of like, how are you conceptualizing how that time course kind of could?
Yeah, I think that's a fascinating question. And, we don't know how common persistent anosmia is going to be. We have some ideas. So there's this there are a number of coronaviruses because they, are common cause of the common cold that have been studied in the lab and explored in terms of their ability to affect the olfactory system. There's actually one version of a coronavirus that you can give to a mouse, and it'll infect specifically sparse populations of these sustentacular cells. And then over time, that causes basically the olfactory epithelium itself on the whole to collapse. And so it suggests that there could be a kind of a significant consequence to even taking out small subsets of these support cells. And if, in fact, the stem cells, which are important for renewing this epithelium when it gets damaged, if they also are subject to infection, it could really delay the recovery of the olfactory epithelium. That's just a hypothesis, right? We don't really know what's going on, but I think that's kind of how we're thinking about it.
Is that mouse hepatitis virus by any chance?
It's a variant of a mouse hepatitis virus.
All right. The bane of mouse facilities.
Yes, exactly. Exactly.
If you get that in your mouse facility, you're in big trouble. Wow. So is there anything you can tell us about what you're planning to do with this, Bob?
Yeah, I mean, we, of course, are interested in validating these results. Again, they're all, much of this is data mining and much of this is based just on sequencing. And so currently, we're doing even though we're slowed down as best we can under these circumstances, we're trying to get some antibody staining going to see what the protein is, doing some in situ to validate what we've been seeing in the sequencing. I think one interesting question is, if you were to take SARS-CoV-2 and you could do this experiment, if you put it into a nose, where would it actually go? And so there's a couple of versions of that kind of experiment one can imagine doing. One can pseudotype a different kind of virus with the SARS-CoV-2 spike protein and then ask, in the mouse, where it goes. One of the issues with doing these kinds of experiments is that the mouse ACE2 is a much less good receptor for SARS and SARS-CoV-2 than the human protein. And the only mouse models we have, at least as far as I'm aware, are mouse models in which the human ACE2 has been transgenically expressed under a keratin promoter rather non-specifically. And so if your interests are in like what cells are getting infected and how things might spread, that mouse model is not an appropriate model to go study. And so one of the things that has to happen if in my lab or any other lab is going to continue this work is that someone's got to knock in a human ACE2 into the mouse locus and generate an appropriate mouse model for us to understand pathogenesis.
Someone told me, by the way, you only need to change one amino acid in the mouse ACE2, and now it will be fine as a receptor. So you don't have to knock in the whole thing, and maybe it's easier.
It's a little CRISPR here, a little CRISPR there, and it solves your problem. Yeah, but until
or use ferrets. Aren't ferrets of it?
Yeah, that's
ferrets seem to be good host.
For a systems neurobiologist, you sound like a pretty good virologist.
I think I'm just a fast learner.
Pseudotyping, it's pretty good.
Yeah. So there's another bioRxiv preprint from another group. I guess you've seen this. Are you aware of this group and their work?
Yeah, and I think that their findings are kind of generally consistent with what we've been seeing. So it's great to see someone else be able to observe the same phenomena.
Yeah. Your paper is beautiful. A lot of data, nice figures and so forth. Hopefully you'll get it published soon.
Yeah, if I can say just one thing about that, which I think is surprised me kind of, and it's just super awesome, is this was our first bioRxiv paper. And when we first put it out, just our group and Darren Logan's group were authors on it. And then when it came out in bioRxiv, we got calls from all sorts of people in the community who had data and were interested in joining up. And so like the current version of the paper, instead of having 5 authors, is like 30, and it's a large international consortium of people who are interested in this question and have contributed lots of unpublished data sets. And so we're really excited about how open science has really facilitated our look at this problem. Certainly any one of our labs, given that we're all shut down by COVID, couldn't have done this. But together it's ended up being a really nice story.
Yeah, I was going to ask, you have all these different sites. You got Genentech, you have Belgium, you have UK. Oh, wow. So these people approached you. That's really neat.
Yeah, it all kind of came together and nucleated nicely after the first posting. It was really great.
So I think this is precisely why I love preprints. And, maybe Bob, you should write a tweet about this, because I think there's always some skepticism from the community about the value of preprints, but this is a prime example. I mean, I guess the other thing that we're seeing is that, well, it's actually possible to have fast peer review, right? Like, it's amazing to see these preprints go into Science and Cell and whatever, and it's been like literally a few days.
Yeah,
I don't know.
We're doing the peer review right here.
I mean, it's a question of how thorough the peer review is, but I think at least it's doable. I mean, oftentimes the delay is from the editors and not from from the actual reviewers.
This is also a really great use of single cell sequencing. I think, one of the things that happens when technology evolves is people just use it just to use it. And so, a lot of times, especially in the neuroscience field, I see a lot of single cell sequencing stuff that doesn't really need to be single cell sequencing, or like is single cell sequencing, but identified cell types we already genetically defined before. And it just kind of doesn't make a lot of sense. But this is such a perfect question for single cell sequencing, especially when you're thinking about like a virus, right? Because you have cell types, you know what they are, but you don't know how many of those cell types may express viral proteins necessary for viral entry. You don't know the percentages across these different cell types. And it's a really nice way to, one, use existing data sets, right? Because a lot of these are freely available, but two, ask a question that actually requires cellular resolution. And so I think it's a really kind of great way to ask this question, and it's accessible. That's the other thing. I think a lot of big data papers are hard to dig through. And so I actually really like kind of your approach here and how you're using it and how clear and easy it is. And I think that should be kind of the goal of these kind of big data sets. How do we distill this into stuff that people can understand and we're using it to answer a question?
Yeah, I agree with Erin there. I mean, the amount of RNA sequencing that's coming out in neuroscience and how much value it has, I'm very skeptical. But this is a clear question, clear answer, and you can validate it later. I mean, I think that's the cool thing.
Well, it's all out there now for us to go back and do these analyses. That's kind of, you never know what will be out there, what will be needed.
Ori, is anything else you wanted to do before we sign out?
I think this was great.
All right. This is our 5th episode. We're moving along. There's this magic number in podcasts. If you get past 12, they're likely to last. So.
Well, and you just got to 600 on the other one.
Congrats.
Congratulations.
Thank you. 12 years. Yeah.
Do you have your sculpture yet, or has it not come?
No, all of you guys chipped in to buy this Luke Jerem sculpture. It didn't come yet, but when it does, I'll make sure to show it to all of you.
Yeah, I didn't know he made. I mean, I'm tempted to see if he can make an arc capsid.
I'm sure he could. Yeah, I'm sure he could. He's really. I was at the University of Maryland, and in the student union, in the lobby, they have one of his sculptures in a case. Someone donated it, so cool. It's very cool. I'll have to donate mine to somewhere, so it's there after I'm gone. That'll be cool. But thank you all for that. That was very nice of all of you to do that. All right, so that's TWiN 5. You can find the show notes, microbe.tv/TWiN. Questions and comments, twin@microbe.tv. You can listen on any podcast player. Please subscribe so you get every episode. And they're all free, of course, and that way you get them when we release them. If you like what we do, consider supporting us. You can go to microbe.tv/contribute. We have a few ways you can give money to help with our efforts here. Today, our guest has been from Harvard Medical School, Department of Neuroscience, Bob Datta. Thanks so much for joining us.
Thanks so much for having me.
Thank you, Bob.
Yeah, thanks for coming, Bob.
Ori Lieberman's at Columbia University Irving Medical Center on Twitter. It's Ori Lieberman. Thanks, Ori.
Thanks for having me, Vincent.
Andres Bendesky's at Columbia University. Bendesky on Twitter. Thanks, Andres.
Yeah, it was a pleasure.
Erin Calipari's at Vanderbilt University on Twitter. The Erin Calipari. Thank you, Erin.
Okay.
Jason Shepard, University of Utah in Salt Lake City. Jason Synaptic on Twitter. Thanks, Jason.
Thanks, Vincent.
And I'm Vincent Racaniello. You can find me at virology.blog. Thanks for listening, everyone. We'll see you next time on This Week in Neuroscience.